Good afternoon. I'm Seong Eun. I'm from South Korea, so it's been 10-hour flight. Yeah, I'm so tired, but I'm really so honored to be here as a presenter. So I'm a senior researcher in the Center for Human Care System at Korea Electronics Technology Institute. I'm going to talk about today development of fluorescent proofs and biosensor platform for cancer diagnostic application. So there are two parts of this talk today. First thing is to fluorescent proofs and biosensor platform, especially for early cancer biomarker. And next one is ongoing project, which is a highly sensitive and miniaturized fluorescent-based biosensor for POCT device. So the cancer is a term used for diseases in which abnormal cell divide without control and are able to invade other tissues. But as all we know already, the cancer is the leading cause of death worldwide. So as the number of cancer cases rises, so does does the cost. So the cancer economic cost gets also enormous these days. So this is why we need to develop a biosensor platform for cancer diagnostics. So we have actually already current standard tests such as tissue biopsy and cytology and cystoscopy, but they have this is really crowded. They have a lot of drawbacks nowadays. So to summarize them, they are invasive and expensive and time intensive and dependent on operator skill and also painful and causing side effects such as injury or infection. And even more, biomarkers, recent biomarkers and receptors are not that sensitive enough. So now we can recognize 4 aspects as below here that we need to consider for developing cancer diagnostic tools. Novel biomarkers, receptors, high sensitivity and non-invasive simple detection and point-of-care testing purposes. So I'm going to introduce one of the examples for novel biomarker discovery. So this is called DNA methylation levels and patterns. This one is associated with specific cancer as listed in this table. It's off. So over here. In cancer cell, as you can see here, they have different methylated DNA pattern compared to the normal cell. And so it's been reported DNA methylation changes have occurred early in carcinogenesis. So at the same time, we need to develop a simple and sensitive biosensor platform for detecting this kind of DNA methylation pattern and levels. So this is general designing principle of biosensors for diagnostics. So this scheme actually have been shown up during this symposium several times. So to detect the various analytes, such as DNA methylation, the probe needs to be developed carefully, consists of recognition elements and signaling development, depending on the type of signal transducer. So this is what I did for developing the probe. So I actually named it as method probe that can detect DNA methylation. So I carefully selected the domain for detecting methyl group on DNA, and then I carefully engineered the protein domain as well and labeled it with some fluorescent molecule. Since I was using like fluorescent-based transducer. So luckily, the development method probe has higher affinity and less non-specific binding than wild-type domain itself. So this is actually the experimental result, like a highly purified method probe over here. And this is the instrumental setup that I use, experimental setup that I use, which is called fluorescent correlation spectroscopy, so-called FCS. This instrument collected the fluctuation of fluorescent signal from fluorescent molecules within this kind of small focal volume over like reddish color over there. Then the collected data can be fitted by some correlation equation. Then we can get the diffusivity of the fluorescent molecule with this FCS machine. So actually this is the key for developing a biosensor platform, since it gives us to make some simple detection schematics. So this is the schematics of probing method that I developed to detect the biomarker. Just simple mixing fluorescent probes with DNA sample is only required in this platform. So as you see this picture, when method probe bind to the methyl group on the DNA, then the whole complex, the methyl probe gets slower. So we can get like slower diffusivity of bound complex. So as increased methylated DNA, then we can get the slower diffusion of the complex compared to unmethylated DNA showing like constant diffusivity. So a simple one-part assay system was developed for quantifying the amount of methyl group on the DNA, independent of DNA sequence context by testing with differently patterned methylated DNA. So finally, the simple assay system that I developed was examined using like several different kinds of DNA sequences as well. So based on this background, I tried to develop some fluorescent-based biosensor for POCT purpose. So this is ongoing project, which is called highly sensitive and miniaturized fluorescent-based biosensor for point-of-care testing device. So now actually we are trying to design some simple detection that can be integrated with in the semiconductor based photo detector. So what do we need for a point-of-care testing device that with the three aspects that I mentioned earlier: biomarkers and high sensitivity and simple detection. So actually the point-of-care testing device are aiming for like robustness, miniaturization, and low cost and digitalized service. since it should be portable and people can afford to buy it for on-site diagnostic, and also the immediate service information, immediate diagnostic or prognostic information would be much efficient way for like POCT purpose. So we think actually the key to POCT device is the currently developed silicon nanowire. In silicon nanowire by us collaborated with SilTerra, the host in this symposium. So as you can see over here, like the upside down triangle over here, this one is silicon nanowire, which is floating on this silicon. So let me tell you about why we think this silicon nanowire is the key. So this is a silicon wire fabrication process, which is top-down method. So it begins with initial oxidation and dry etch and wet etch, and lastly, thermal oxidation. Then we can get the silicon nanowire as described in this picture. And so our developed top-down method process allowed large scale fabrication and high reproducibility and mass production. So this picture describes like the top-down method that we set up. And so it's, but however, like the currently used VLS method, which is top-down method, is just giving like low reproducibility and difficult to mass production. And the developed nanowire array has high optical responsibility. So due to the high gain mechanism of the silicon nanowire, which is not, I'm not explain detail today, compared to the commercial one, which is silicon avalanche photodiodes over here, our developed one shows really higher optical responsibility at low bias voltage. It means it will give us like low powering. And also it has even high optical responsibility, like at 400-ish nanometer wavelength, which is wider. And in addition to that, it's way cheaper than the commercial one. So to develop, like simply design the POCT biosensor device using silicon nanowire-based photodetector, we are designing like simple detection platform which can be integrated in with the silicon nanowire-based photodetector. So now we actually expect that we end up having like a cheap and robust and miniaturized POCT device at the end. So now I feel actually it'll be realized very soon, since like many experts here like sitting in front of me. So yeah. So thank you for your attention. Yeah.